cell enters into the cell cycle. The cell starts off in the G1 phase of interphase, where the cell will grow larger and larger. As it grows, it copies its organelles. Next, the cell enters the S phase, where it copies all of the DNA inside of the nucleus. This means that each of the 46 chromosomes are duplicated by the cell. After this, the cell enters the G2 phase, where the cell keeps growing and starts prepping to divide during mitosis. Now the cell enters the M phase, or mitotic phase, where the cell will divide its newly copied DNA and cytoplasm into two daughter cells. To undergo this, the cell must complete mitosis and cytokinesis. Cells that don't divide or are stopped in the process rest in what is called G0. Cells that rest here enter after the G1 phase. They do not prepare to divide until they are given the right signal, or in some cases, they are pre-programmed for cell death. Cells that go through apoptosis or cell death are cells that did not pass the checkpoints during interphase and as a result were sent to G0. Checkpoints are located after the G1 and G2 phase. They make sure that the cell is up to speed with the cycle and not engaging in abnormal activity. Cell cycle controls. The first cell cycle control is a cyclin. There are 4 types of cyclins: G1 cyclins, G1/S cyclins, S cyclins, and M cyclins. Each particular cyclin is associated with their particular phase or transition. Their role is to promote the activity of their specific phase. Their levels of concentration vary across the cell cycle, but typically they accumulate during their phase and lessen in concentration after. The other cell cycle controller is called a CDK. These are enzymes that speed up actions in a cell. CDKs, or cyclin-dependent kinases, are very plentiful in our bodies and are simply dependent on cyclins. When a cyclin binds to a CDK, it activates it, allowing it to become a functional enzyme and modify target proteins. This process is called phosphorylation. In basic terms, the cyclin attaches phosphate groups to specific target proteins, and the attached phosphate group activates the target protein. Once a cyclin binds to a CDK, it not only activates the CDK, but also guides the CDK to specific target proteins. Another cell controller is an MPF, or maturation promoting factor. When enough active CDKs are built up, they can help the cell begin dividing. MPFs are activated after the G2 phase and assist cells in entering the M phase. MPFs trigger the formation of the spindles of the poles of the cell, which pull apart the chromosomes. Mitosis is the dividing of diploid cells into identical daughter cells. Diploid cells have 46 chromosomes. In order to divide, the cells must copy all of its DNA. Its division occurs through the following stages. Interphase is the first phase of mitosis, where the cell is still whole, the nucleus is distinct, and chromosomes appear in the form of chromatin. Chromatin is long and thin, it resembles a piece of string. Prophase is the second phase. In prophase, the cell appears whole and the chromosomes become clear and condensed. The third phase is metaphase. During metaphase, the chromosomes are lined up in the middle of the cell. Spindles located at the poles of the cell are attached to the chromosomes, ready to pull. The fourth phase is anaphase, in which spindles have started to pull the chromosome towards the ends or poles of the cell. This is the first stage where division becomes distinct. Fifth is telophase, where the chromosomes have been split and pulled to the poles. The cytoplasm divides, the cell begins to split. Lastly, there is cytokinesis, where the division is finished and there are 2 daughter cells. Each daughter cell is identical to the parent cell. These cells will undergo mitosis again, creating new cells as the process starts over. Meiosis is very similar to mitosis, aside from the fact that cells undergo 2 divisions instead of 1. Meiosis deals with sexually reproducing cells. The cells that undergo meiosis are haploid cells and they have half the number of chromosomes. Meiosis begins as well with interphase. The nucleus is distinct and chromosomes appear in the form of chromatin. The cell then enters prophase 1, where a process called crossing over occurs. Crossing over is when sections of the maternal and paternal chromosomes are switched into the daughter cell. The interchanging of chromosomes causes genetic variation. The next phase is metaphase 1, in which pairs of identical chromosomes, called tetrads, line up in the center of the cell. The chromosomes, like in mitosis, are attached to spindles at the pole of the cell. Anaphase 1 is the fourth step, where the tetrads separate and spindles pull them to the poles of the cell. Fifth is telophase 1, where the chromosomes have been split and the cell begins to divide. Lastly, for the first set of phases is cytokinesis, where two daughter cells have been created. Each of these daughter cells has half the number of chromosomes as the parent cell. These two cells then enter meiosis 2, where prophase 2 through cytokinesis 2 are completed. Each of these stages is a repetition of the first. Eventually, 4 haploid daughter cells are formed. Each of them will have only one chromosome from the original parent cell.